So my name is Jagdeep Walia. I'm from Queen's University in Canada. And thank you, Dr. Baker, just you introduced me. We are going to talk about GAMT gene therapy that we got durable and dose-dependent response today. I hope that everybody is awake after lunch. So my disclosures, I'm a consultant with Life Labs and DynaCare and some consultants as well. So what I'm going to start with is with some background, but we have got enough background from Dr. Longo and multiple other speakers, so I will not go for a bigger background description of what creatine deficiency disorders are, but I will go straight to the study design and the results which we need to share, and then we'll have a discussion about that. So all this work is done by my students. The girl who worked hard on this, she is a PhD student. Her name is Robin, and I have another student who's coming next. His name is Troy, but I'm just presenting the huge amount of work she has done. So we know about the creatine as an energy buffer. So let's forward this because we know about the creatine biosynthesis, what it does in the brain. So there are two compartments. So the whole body is one. But because of the blood-brain barrier, now there are two compartments. There are two compartments, and both of them are equipped to make its own creatine. So you make creatine between liver and kidney, which is good for peripheral circulation. Some of it goes into the brain. But because of blood-brain barrier, not much of creatine goes into the brain. And brain developed its own enzymes to create its own creatine. And that's why it's very important that whatever treatment we give, it has to be brain targeted. So this is the GAMT deficiency. Main things are two. One is that there is guanidinoacetate, the GAA. It is increased in GAMT deficiency, and there is no production of creatine. So that's what we are going to focus in my presentation today, is that how can we decrease GAA and how can we increase creatine in GAMT deficiency by using gene therapy. So we are using an AAV9 vector. It is expressing GAMT using a ubiquitous promoter, and we are giving intrathecal injection. So it is an LP, and we gave it in mice at around 6 weeks of age. So Robin gave the proof of concept study description and presentation last time, and it was an online presentation, virtual conference. I'm going to present what we did next to it. So what we did was that last time Robin showed that she chose one dose and we injected, and it was a wonderful increase in creatine and decrease in GA. And what we wanted to do was to see what is the best dose which can be given, and it will create a good amount of creatine and GA reduction. So we chose multiple groups, multiple mice, and we chose multiple doses, increasing almost double every time. So it's from 6.25 E10 vector genomes, and then it kept doubling every time. Because for all gene therapy projects, we give a combination of prednisone and rapamycin, and the whole idea is that we don't know that how much immune response will be created. It's a one-shot gene therapy. We want to create immune tolerance. So we start with prednisone and rapamycin for almost 4 months, and slowly taper prednisone, and then we measure the enzyme levels and the GAA. So there'll be 2 time points. One is going to be called short-term endpoint. So we sacrifice some mice at 13 weeks, and then long-term endpoint when we have finished the immune suppression regimen. And then after that, almost 1.5 months, we sacrifice the rest of the mice. So the results. The short-term result, which when we sacrifice mice at 13 weeks. So if you will see, this is an important part here. So if you see the creatine in serum, in blood, what did we find? We find that the creatine went up in high doses to almost normal. So the black line is the heterozygotes, and as compared to heterozygotes, if you see that these three lines, they almost reach the normal heterozygote levels. And similarly, GAA, which we need to reduce, so if you see this black line, which is again heterozygotes, they have almost no GAA, and when we gave in the higher doses, they almost reach the heterozygote levels. And this is the final 13-week slide in bar graph. So if you see this, that as the dose increased, there's a dose-dependent increase of creatine in the blood. But more importantly, you will see that the amount of creatine made and amount of GAA which is reduced is almost same as heterozygote levels. So we reached what we wanted to, but what we saw was that if we go to higher doses, so after the medium dose, if we go to higher doses, there was no significant increase in creatine, and there was no significant decrease in GAA. So there was some kind of ceiling effect after one dose of 2.5 E11. So, and this will be reflected more as I go ahead and give more presentation. So this is important to note that after the medium dose, there was a ceiling effect. There was nothing happening after that in clinically or biochemically. So some people were saying that did we do any male and female analysis as well? We did a behavior test, and we found that there was an increase in mesh stressed strength test as the dose increased, if you see this way, at 8 weeks as well as 12 weeks, and it was similar in males and females. All right, so let's look at the brain, because serum is a cumulative measure of how much GAA or creatine is there. It is from different organs and brain. So we started looking at different organs now. So in brain, we saw the same thing. We divide the brain into 3 sections. So there's a rostral section, midsection, and caudal section. And we divide that, and then we analyze all the 3 parts. So if you see this, the creatine levels increase with dose in all sections. So as the dose increased, it kept increasing to a point of the mid dose, and then after that, there was no increase in creatine, and it is happening in all the sections. So there was no significant increase after certain dose. We look at the same thing with GAA, and if you see that there was a dose-dependent decrease in GAA, and it kept going down as the dose increased, but after certain dose, there was no further decrease of even GAA as well. We looked at the spinal cord, and we looked at the peripheral organs. So even in the peripheral organs, it's the same story, that, for example, in muscle, there's a dose-dependent increase in creatine in liver as well, and as well as kidney, but after the certain dose, there was no effect going on. So we wanted to know that, and same thing with GAA. So GAA in the muscle, it was high in knockout, so this red line is knockout, but as the dose dependent, there is decrease, so there was almost no GAA coming out. That was the best result we got from muscle. And then there was in liver also the GAA decreased, and in kidney also. It decreased slowly in the higher dose, but then didn't go further down. So then the question was that how much, was there any actually, was it our virus went wrong after one dose, and that's what the reason? But that was not the case. The copy numbers actually kept increasing with the dose. So if you see this, as the dose increased, the copy numbers increased in different organs. So it was not because the virus is not going in different organs. It is making, the copies are there, DNA is reaching all the different organs, that is whether it is brain or liver or muscle, but there was no clinical biochemical effect happening. The second thing from this figure, which is important to see, is that you don't need a lot of copy numbers in different organs to make that biochemical effect of correction. We reached almost wild type or heterozygote level of correction, but you don't need a lot of copies. So if you see 1 in 10, 10% to 5% copies in the brain were enough to create the effect, which is required to correct this disorder. So, so if you see there was the increasing dose, and I just pointed out that only 10% to 5% is enough to correct this disorder. So from this study, in the short term, what we found was that in the serum, the creatine and GAA normalized. But in brain, it was normalized for creatine, but not for GAA. There was still some residual GAA in the brain. But we reduced it to 80 to 90%. We increased the dose, increasing the dose or expression of the of the gene, where I use the same vector, AAV9, using the same IT route of delivery, it will not increase the biochemical effect in brain. So that's a very important conclusion to say. Even if I change the promoter to a strong promoter, it's not going to change the biochemical effect, because there are only certain cells which are transduced, and IT has a certain way of distribution in the cells. So even if I say that I increase the dose, that means the virus is going in the same areas. The copy numbers per cell may be more, but it's not changing the biochemical effect. Similarly, even if I put the strong promoter, and if the amount of enzyme being pumped is more, still there will be no biochemical effect, because overall the gene, there is a ceiling effect. Now the residual GAA means there are still areas of brain that possibly has higher AGAT to GAMT ratio, and this is my thinking, that the AAV9 has gone into certain parts of brain, it is changing the AGAT to GAMT ratio, and that's why the GAA is getting consumed. But there may be some areas where AGAT is sitting alone, poor guy, and is pumping the GAA, and the GAMT is not there to help it. So that's the question which comes is, do we need to make GAA completely normalized in brain? And that's what I asked Dr. Longo just a few minutes ago. And he said, no, if 80 to 90% of the GAA is normalized, then you don't need to go any further down. Is that right, Dr. Longo? Plasma is normalized. Yeah, so, but brain is more important part here. And it's maybe that the GAA needs to be used for something else. Anyway, in long term, so there is a durability, whether this effect was durable. So we wanted to see this, and we saw that actually it was durable. The heterozygotes showed increased creatine with time, but also did our mice. As the time passed, there was increase in creatine, and the GAA was kept at low level. So what we saw was that the creatine levels kept increasing with age. So if you see the levels, it was around 75 at 16 weeks, and it became like almost 100 at 26 weeks. And the GAA was almost similar. And then we looked at the brain. So still at almost 6 months of age, these mice, so we just continued with only the 3 doses. We didn't continue with the last 2 higher doses. So we see that the effect is still continuing the same with increase in creatine, and there was a decrease in GAA. And we see the same thing in muscle and liver as well. Our conclusions are that the vector which we are using significantly improved the phenotype of GAMT mice as compared to the untreated animals. The effects seen following intrathecal treatment were dose dependent, and it was durable, and up to the 5 months or 6 months of age, till what we kept the mice. So we finished the proof of concept study, we finished the dosage study, we know that which dose we can identify which can be clinically translatable. Currently we are working on immune response studies just to see that if there are any immune responses, and if a regulatory agency asks for ELISA and ELISpots, then whether we can create that assay. And then what we are left with a toxicology study, and we're good to go for clinical trial. And also, funding. And I want to acknowledge that this work is not done by me. I just supervise, but there is a whole team which works. Robin is there in the center, who has done most of this work. And there is Troy here, who is going to be presenting next for creatine transporter deficiency. With that, I just finished. Thank you for ACD for letting us come here and present our data. And thank you all the families and patients here as well for listening.
Okay, great talk. Any questions? Your graduate student would like Any other questions outside of the Walia lab? Yeah. Oh, yeah.
So it seems pretty definitive. What would the timelines be for the third and the fourth stages before we get to clinical trials?
So usually toxicology study should be for 6 months. And then you need to make the vector. Vector production takes anywhere from 6 to 12 weeks. So let's say 3 months of vector production, 6 months of toxicology study, and then we are good to go. But before that is funding. So that can take, I don't know how much time.
And the administration, does it have to be through the spine, or how would it be administered?
Yeah, it will be a lumbar puncture intrathecal injection. Okay. And I think that's the easily clinically translatable method if animals have, or preclinical work has shown it will work. For some diseases, it may be not good, and for some diseases, we may have to switch to ICV method. But for this disease, lumbar puncture works wonderfully well, and it's easily clinical translatable.
Thank you.
Great. And I'll ask a follow-up to that. I thought this was fantastic. I mean, the efficacy in terms of creatine and reduction in GA is awesome. Just to sort of make sure that I'm clear on what the model is the difference in copies of the vector between the lumbar and the cervical spinal cord statistically different? Do you think that the cervical spinal cord has a lower number of copies of the construct than the lumbar puncture? Or yes. The lumbar spinal cord?
So wherever you will put, let's say, 100 vector genomes, so 100% virus is delivered to lumbar puncture, that will absorb most of the virus there. Yeah. And then it will slowly go to the canal, to the brain. So a lot of virus gets absorbed on the way. So wherever there is injected, there will be higher number.
And then do you think, I mean, there were some copies clearly throughout the brain, but certainly an enrichment at the lumbar spinal cord level. Do you think creatine is diffusing from areas that are sort of more distal, or do you think it's important to get copies into the higher portions of the brain to diffuse creatine to those structures more rostral?
I think creatine is required by almost all cells, mostly with neurons, so I think the copies need to go everywhere. But the good thing is GAMT and AGAT, we don't need all the cells to express both the enzymes. So if some cells express AGAT and some cells express GAMT, which has been already shown in literature, then they are good enough. When I looked at the literature in gene cards, if you see the distribution of AGAT, it's very well secreted enzyme, and it goes out from mitochondria and cytosol into extracellular fluid. So similarly, GAMT, if you see the gene cards photograph, even though the expression is more in cytoplasm, but there is enzyme in extracellular fluid as well. So there is some kind of cross-correction going on. Is some cells which can produce the enzymes can help the other cells.
Yeah, I bet it's a nearby neighbor helping effect, which is very cool. It's nice. It's awesome that works.
Yes.
Yep.
I'm not very familiar with mice, but the concentration of guanidinoacetate in plasma seems pretty high. That was in plasma, is that correct? 100 micromolar, or was in the CSF? The concentration that you show of guanidinoacetate, is that in plasma or CSF?
That's in plasma.
So usually in human, the normal is, depending on age, 2 micromolar, 3 micromolar. The abnormal that you find in GAMT is 15, 20 at diagnosis and then goes down. But here it looks to me that you have about 100. Is that correct?
That is correct from what it is reported from Dr. Andreas Schulze lab. So we take the samples, we send it blinded samples to Dr. Schulze lab. There is a technician who analyzes it, sends us the values, and we put the numbers back. So it's totally blinded game, and the numbers come from his lab.
Thank you.
Steve.
Thank you. How much funding do you need?
Ideally 10 million, but we can start working from 5 million as well.
We got some fundraising to do.
That would be very helpful.
I have a normal person question. You were talking about the creatine. When you gave more, you didn't see a change. How would that be with us just administering creatine to our children daily? Are we saying not giving more is not going to affect them? They can only store so much and help them.
So I didn't say about creatine. It was a virus which we gave. So the virus which has the transgene, the dose has a ceiling effect, not the creatine. So the virus, when we keep giving at a higher dose, it reached a ceiling point. It plateaued after 2.5E11 dose. But that does not change if you are giving oral creatine powder. That's different.
Okay.
All right.
Thank you all.
Thank you.